Okay, so we'll get started. Any questions from the last class or anything that was not clear? So I will be releasing the assignment this evening. It will be due exactly 1 week from when it is released. So if it is released 6 p.m., it will be due 6 p.m. next Monday. It's mostly straightforward. This requires some calculations. Uh, People who have people written LaTeX. Are people familiar with LaTeX? Want to just level up if you haven't. Well, you can submit your answers, handwritten answers also, but no hand submissions, no in-person submissions. All of this will be through a website called Gradescope. I've already created the account for you, so you would have received an email. Everything will be through that. No paper submissions, none of that stuff. Okay. I will give you a template to write your answers in. I will give you Markdown, a a Quarto template which I had shown in the class, but also a LaTeX template. You can choose whichever works for you. There is also a coding question for which I require a PDF submission. I used to allow submissions using Google Colab, but people again started gaming the system. So if people game the system, I have to take a step back. So only PDF submissions. Your code should be visible, your output should be visible, your conclusion should be. If you're using Jupyter Notebook, your answer should be in the Jupyter Notebook, compiled into a PDF. If you cannot make a PDF, take an HTML, print it as a PDF, and attach it one PDF submission for for the entire assignment. Okay. So it should not be very difficult. First, this is going to be first, so I think there will be some hiccups, but hopefully that's the first and the last time we have a hiccup, and then things move smoothly from there. Okay, so people who just walked in, I'll release the assignment later today. It will be due a week from now, and it's right now it's theory questions, one coding question or programming question. I'm not looking for your code as such. You can choose whichever language works for you, but I want to see the code. So it cannot be, it cannot just put a PNG or a PDF or a image and your answer. I should be able to see the code just above it. So you can use Google Colab, you can use R, whatever language you want, I don't care, but it should be visible, like your code should be visible, whichever language you use. R, Python, C++, Julia, whichever language you want to choose. For the theory questions, it would be a good idea to learn about LaTeX. I know it's asking too much, but you know it will pay off. It's one of those things which will pay off in the long run. So it's spelled like this, but it's called LaTeX. It's a very simple thing, and now you have access to all sorts of LLMs, so you know, this should not be very difficult. Used to be a huge steep learning curve back in the day, but now I think that learning curve is all gone. These slides, by the way, are also written in LaTeX, for example. So that is, it makes writing math easier. If you don't want to do it, that's also fine. You can take an image of your. So if you're doing a derivation, you can take an image of that assignment, upload it as a PNG, and still submit a PDF file. You want to write answer in doc, that's fine. At the end of the day, whatever you works, uh, I will only allow one PDF submission because the interface only allows PDF submissions. Okay. Okay, so we'll start with learning something about Stan Ulam. Anyone here has heard about Stan or Ulam? Yes, no. He's a Polish mathematician, played a fundamental role in Manhattan Project. People have heard about Manhattan Project, right? So this is he played a seminal role in of course in Manhattan Project, which is of course not the topic of discussion here, but he also gave a seminal talk in one of the biology conferences where he introduced the idea of biometric spaces, and he proposed, this is a full-on audience of biologists, where he's proposing that rather than biology asking mathematicians to do something, why don't you, um, rather than asking people, rather than asking mathematicians to do something for you, there are avenues for you to do something for mathematics. So what we will be seeing today, which is an alignment problem, which I motivated in the last class, came from some of those ideas. Of course, in transition, some of those ideas have not pulled up, but the idea of laying down foundations of how to do comparison between two sequences can be attributed to Stan Ulam. Okay. So, those days, mathematics and biology were like used to talk to each other. Unfortunately, our education system has forced it to take into two different routes, which actually is problematic. And of course, some of you, those of you who have decided to stay on, hopefully relate to this idea that we need to break those barriers. Math cannot exist in isolation, and so does biology. Like biology cannot exist in isolation. Uh, Mathematicians should not be afraid of biologists or biology. People who are doing biology should not be afraid of maths, right? It's at the end of the day, everything is just logic, right? And your mind is designed to be able to logic, right? So if you can logic, if you can come up with logic, should not be afraid of any subject, right? So why am I talking about this? Because this class started, as I told you, this is like your child. So this course is my child. And those of you, if you know, well, none of you have a child, neither do I, but you'll relate hopefully to this idea that sometimes parents get troubled because the child wants to play with everyone, but not everyone wants to play with your child, right? So this is example of that course. I started with a lot of ambition for getting people interested in it. 63 people showed up in the first class, I think, and now it's down to 39. So something I'm doing very, very wrong, or there's some essence which I've still not been able to crack. So anyway, I will keep trying, and hopefully someday this institute will see more people not being scared of this field in general. But thank you for those of you who have stuck around. Hopefully you have stuck around. Okay, so we'll begin with this idea that we are interested in finding out. I've identified a new gene, which means I've identified a new DNA sequence, and I want to understand what function it carries out. Now, I'm not the first biologist in this world, so there are other people who have likely studied biology and who have taken or who have studied some form of this particular sequence in the past. So rather than trying to do a functional validation, what is a functional validation? So I identified a new gene in a particular organism. I would try to do functional studies with it. So I will go to the lab, change the sequence and understand what impact does it have on the final output. So if I change a particular DNA sequence in this particular input sequence, let's say this is that sequence that I identified in the lab, if I want to understand what function it carries, right? The approach that we take is, we can take this approach of single site mutagenesis. So we will go base by base. I will say that, okay, if this sequence is very important, what happens if I change it to a different sequence? And then I will do an experiment and I will try to measure. This is an arrow which is usually used to depict whether a gene is on or off. So I change this particular sequence and I see that what was on earlier is now off, which would tell me that this sequence is likely associated with some form of a gene expression pattern, right? So this is ACTGA, this is CCTGA. However, doing this experiment in the lab is very costly, and also it's very time consuming, but also I'm trying to reinvent things which would have otherwise probably been studied by now. So many people have come before me, they would have done some experiments with maybe somewhat similar looking sequences. So this idea can be, I can make this approach faster by inferring, by trying to find out what other organism or what other sequences have people studied and what are their functions. And people would do this kind of experiment early on, like 1970s, 80s. People would actually do functional experiment, and then in 1990s and so on, a new algorithm came about to be, which is what is now colloquially used. It's this algorithm called BLAST, and we will see what does BLAST stand for and how does it work today. But the central premise of any of these algorithms, right? So this is a molecular biology problem, but now we have flipped it apart and now we have made this a computer science problem. So last class we were doing statistics, we will do some computer science, we'll try to do some dynamic programming here. The idea is, if I can take the sequence and understand in which other organisms does it exist, and has somebody annotated the properties or the function of the sequence, right? So for example, this is in, let's say, frog, and maybe in human, if not exact, but a similar kind of sequence exists. I could say that, okay, I don't know the full sequence, but there's a very similar-ish sequence which exists in the frog database, in the human database. So maybe this sequence is also carrying out a similar-ish function in frog as well. That's the premise on which this particular tool of alignment works. Is the central idea clear? But why? Why should I make that assumption? Why? I gave you this assumption, but why? Why does it make sense? Does it make sense at all? This is the sequence in frog, and I'm saying that, somewhat similar-ish looking sequence, or maybe the exactly the same sequence found in human, would likely be carrying out a similar function, if not exactly the same function. Why does that argument make sense? Does not make sense? Why should? Mhm. Sure. So if we knew something about these two DNA sequences, but why should similar looking things have something similar in terms of their function? Right. So they would probably code for same proteins. That is one possible likelihood, that maybe I did not tell you, but maybe this comes from the protein coding region, which makes the protein. So it's quite likely that if the sequence is similar, I can end up making a similar protein, and if I'm making a similar protein, then it's likely going to carry out the same function. That is the premise on a lot of which initial work on DNA sequencing and DNA alignment was based on, and that premise still holds. Other way to think about this is different species originated from one common ancestor, and when there was one common ancestor, there was a choice that that ancestor, when the bifurcation happened, right? So from human, let's say, I asked this question early on, right? But I did not tell you. Right? So if I take a very simple example of human and mouse, around 90 million years ago, they shared a common ancestor. And then some split happened. When this split happened, there were certain new DNA sequences that became part of human and then became part of mouse. But if a particular DNA sequence is still there in these two organisms or multiple organisms, it has to be, it has to have some meaning. There's no reason otherwise it would likely just stick around. There's always a reason why it can stick around, because there is of course uh, random mutations that happen, but the chance of that happening in a very long sequence is very, very small. So this central idea is called that if something is constrained, right? So the sequence is constrained, it likely also carries out a special function. It carries out a similar-ish function across species. That is the entire premise on which this alignment algorithms, well, the alignment algorithms work because thanks to computer science or thanks to some mathematics. But this is the relationship to biology. It's not just random noise. Yeah, sure. So if I take human and all of you answered this question correctly in the first class, which is, it's basically a permutation combination of these ATCGs, and there are 3 billion of base pairs. However, it was not always, this sequence at this particular location was not always like that. If I go to its ancestor, right? How is an ancestor defined? We'll skip in this class, but I can think of this as our forefathers, 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 four parents, four parents, four parents, like 15, 20,000 generations early on, right? So their DNA sequences would have looked slightly different. They could have looked maybe like this. Okay, so there is this part which is maybe similarish, but this part was slightly different. If I now go to mouse, I would probably still see similar. Let me change this. So this is a smaller example, but you can see, and this is a very cherry-picked example, but you can see this particular part has remained constrained. It could have been any other DNA sequence, but it decided to stay as it was, right? As it was from here. Because likely if there is a change in this particular part, it will lead to, it might cause the first quiz question that I asked, which is, while this is what we are able to measure, if we are not able to see this, maybe the organism is dead. That's why it didn't survive. So these processes, which are essentially how the recombination happens and how we infer or how we get DNA mutations as two people sort of give birth to a child. There are mutational changes that happen, but there are also random mutations that get incorporated as we go along, right? At some point, these mutations in a particular progeny will become so many that they become kind of two different species altogether. That's what has happened with human and mouse, for example. But at some point, we all started from the same ancestor. This was like 100 million years ago. So 100 million years ago, if I go back, I would have been able to find a person or a living being which would have similar-ish DNA as compared to today's human and mouse, yet different from what it looks like today. So if this particular sequence has remained constrained, this is constrained because it has not changed, even though it has chance to. There are random processes that will change the mutation. If it has not changed, that means it has to have some biological function. It's not just sitting there around. Which is not true for some of the base pairs. While we don't understand most of it, there is somewhat more importance to certain regions in the genome as compared to the other ones. This one in particular, for example, is a start codon, right? So if the start codon for a particular gene and the corresponding nearby sequences have remained exactly the same, or very similar-ish, it has to have some biological need. Does that answer your question? Okay. Okay, so why care about this, right? So this is an interesting example which I talk about usually. So this, what is an oncogene? An oncogene is a gene that causes cancer, and it was first, well, one of the first examples of this gene particular being studied is in a virus called simian carcinoma virus or simian sarcoma virus. It causes uncontrolled cell growth and leads to cancer in monkeys. So if you take this virus and you infect monkeys, it will give them cancer. We, for a long time, did not actually understand how do humans get cancer. And one of the hypotheses initially early on was somebody took the DNA sequence of this virus and ran a match against the human genome. And they found that one of the genes in humans, particularly the PDGF gene, and again, the name does not matter, has certain sequences that match with the virus. This led to this hypothesis that since we share a DNA sequence with the virus, one potential reason why humans get cancer is because there is something wrong going on in that region where we share the sequences with the virus, which actually is known to cause cancer. Right? So we'll see whether, in the later part of the course, we'll see how much of it is true and how much of it do we know about cancer right now. But this was the example of the right gene. It's the right gene, it's otherwise functional, but it can be made to do the wrong thing because being at the wrong time if there is a mutation that gets incorporated. This was the hypothesis. We'll see how we study such a hypothesis like that in just a bit. So if you take any DNA sequences, there are different possible changes that can happen, right? So if I start from, for example, my DNA sequence, and if I take your DNA sequences or your DNA sequence, in an ideal scenario, it would look exactly the same. However, in a theoretical scenario, it can encompass changes which look like a substitution, an insertion, a deletion, a duplication, an inversion, so many terms, right? Don't worry about it. But what are the possibilities that can happen? Let's say this is your DNA, this is my DNA. This is the first possibility. What changes can happen between yours and mine? Broadly. So this is one example. What can happen? So in a case of a substitution, I can start with this exact sequence, but maybe somewhere in your DNA, I could end up substituting a C with a G. This is a substitution. Okay. You can also have a deletion, which I will not draw the first sequence now, but if I were to draw the second sequence, I could get a sequence where I have a C, but you don't have anything. Everything has been shifted. That is called a deletion. This is a deletion in you. You can also say that, well, I am not the reference, you can be the reference, and in which case, this C is an insertion in place of this, right? So what is an insertion is a deletion for the other person. Is it hard for you as well? No. These ACs are not working. Okay, so one way to other why you see this is graphically. So this is what your normal DNA looks like. A substitution will lead to a GAA becoming an AAA. And what implication this has is it has implications in terms of what, if this happens to be in a region which is going to code for proteins, you will end up either synthesizing a shorter protein or you will end up synthesizing the wrong protein. Okay, we haven't studied the genetic code yet in detail, so what will happen does not matter, but this is what a substitution looks like. You could also have an insertion, so this AAG remains like that, but I end up inserting an extra base pair here, which leads to a second, which leads to the entire thing shifting by one. And similarly, I can have a deletion, which would end up removing that particular thing here from all of this is in this direction. This one is in this direction. This one is in this direction. So the shift, if I insert it here, everything changes downstream, of course. And if a deletion happens, it gets removed from here. So everything moves left by one. Okay. Those are broadly at the base level, the kind of things that can happen. You can have a substitution, an insertion, or a deletion. If you have an insertion in one, it automatically means a deletion in the other, and vice versa. You could also have longer changes, which unfortunately we will not have time to cover about algorithmically in this class. Is you could have certain segments. This is your chromosome. This is, while this is happening at the single base pair level, this can happen at a much segment level, which is an entire segment of, let's say, maybe 1 kilobytes, right, or 1 kilo base pairs. This entire thing can be duplicated. So I can have that entire thing duplicated once more in my entire chromosome. I could also have it as a deletion. So this entire segment gets deleted. This happens, by the way, a lot in a lot of cancers, for example, you either see amplification or duplication, which means that one particular part of it gets amplified. If this gets amplified, what is the implication? What happens? It has gotten amplified at the DNA level, but what implication does it have? Louder. Yeah, lot more expression of the protein, right? So if this happens to be in the protein coding genome, protein coding part of the DNA, then while you were making one copy of the RNA, you will now make two copies of the RNA. And not every disease happens because there is a mutation. Not every disease happens because there is a change is happening in the protein. But the amount of protein that you're going to synthesize, if that changes, then also you can get a disease. Okay. In fact, a lot of diseases happen because of the other reason, which is not because of mutation in the protein coding gene, but mutations because you're not making enough amount of it. Either you're making more or you're making less. That number hovers around a lot, but 90% of human diseases that we know as of today happen because these mutations happen to be in regions which are actually not coding for proteins, right? So while studying proteins is great, and you should take courses in proteomics and so on, there's a lot of change that happens outside these regions. Okay, then you can have an inversion. So what was orange and blue here can get inverted, and you get a blue and orange here. Implication of this is what protein you're going to make. Now you're not going to make that protein. It also leads to, if it happens to be in a region which is not going to bind, which is not going to make proteins, this can change how many copies of your RNA we make. We'll see this slightly later in the class. And there's something called translocation. So these are two different chromosomes. This arm can get translocated to a different chromosome. That also happens in a number of diseases. Particularly, a lot of genetic disorders that begin at in newborn babies happen because of translocations. Okay, any questions about this? This part is clear? So this is just to clear the terminology, and now we'll start to do some calculations. So again, we'll define an alignment using this terminology that if I have, you've already defined it in some sense, that it is defined for two sequences, two or more sequences. You write it by writing one sequence on top of the other. The only constraint is if I have a deletion, then I show it by dash. Does not mean that every time a base should match. This is also allowed, and any other possible combination is also allowed. What is not allowed is I cannot have two deletions. So this is denoted as a deletion. I cannot have two deletions aligned to each other. We are aligning one base at a time in a given sequence of length n. Okay, is the overall notation clear? We will work with once we insert the dashes, right? So the final length of the two strings after inserting the dashes should be the same. You should be able to fit them in a box, okay, rectangular box. And we'll begin our problem with what's called the global alignment, which I'm going to explain. But let's look at this problem first. Logic, pure logic, no math, nothing required, only pure logic, okay? So you have ACG, TC, TAG, and you have another sequence called ACT, CTAG. I have come up with one way to align these sequences. Can you tell me if you would want to align this slightly separately, and if so, how? Okay, so I'm just going to write the sequence first. CG. Okay, yeah. How should I do the second alignment? Okay, AC. Okay. Anyone else? Anyone else? Is that the only possibility? Anyone else? No other combination possible? Why should I, like, why should I, which one should I choose? This one or that one? And why? Any other alternate answers? Okay, let me show you another way to do this. I could also have shifted, started with a alignment here, would have resulted in two mismatches, but would have resulted in a perfect alignment downstream. So shift entire thing from here and let this alignment work out. Which one is better? Like, which one should I choose and why? What's her name? Sirisha. Yeah. Mhm. Why? Why? Okay, so intuitively you all see that, you know, if there's no mismatches, then it's good. But yeah. Hmm. Okay, so that's a biological reason. That factors that if you have a lot of matches, then anything that can actually change the function, you're minimizing for that, right? So that is true. So one idea there is, if I want to quantify this, right, which one is a good alignment, I need a scoring strategy, right? So you need a way to score these quantitatively before telling which alignment is better, and that's what we will do today now. Actually, before we do that, I have a question to ask. We will do that in just a bit. So this is, let's say, this sequence is of length m and this sequence is of length n. How many possible ways, counting problem 101, how many possible ways are there to align these two DNA strings? Length m, length n. Now you understand an alignment. An alignment is basically defined by either this or this or this. Substitution or a match. This is I, insertion in the second sequence. This is deletion in the second sequence. But you can see that they are basically mutually exchangeable in some sense. How many possible ways can I put two strings of length m and n in this fashion? We'll do this intuitively. Question is clear. Actually, the question has to be clear first. So for example, you saw that I could have written, you know, at this point, don't worry about what is the best score, but I could write an alignment which would look like this. I could also write an alignment that looks like this. What did I miss? C, T, C, E. Whatever, there's one extra base. Basically, I can shift everything, shift here, shift there. How many possible ways are there? This is counting problem. Huh? Hmm. Okay, correct. How many? So one thing we had discussed is, it doesn't matter whether I'm doing this also. At the end of the day, I need to be able to put them in a box. Their both length should be equal. Okay. Intuitively, can you see how many places to go? Like, what are the possibilities? What is the longest stretch? What is the longest possible box that you can draw with that constraint? Huh. Max of m and n. Anyone else? Close but not correct. What if I draw this is m, this is n, so I can go till m plus n. Does everyone see how? You can totally put them at the corners. Okay. Okay, what do you do then? That's the maximum length I can go. Okay, fair enough. Edge effects I always ignore, but okay, fine. Hmm. Okay, is that correct? Yeah, they're the same thing, right? So when I write on the top and the bottom, right, they are one and the same thing whether I write bottom and the top. So that part is covered in there. Okay, how many number of possible places I can choose is m plus n, because I will fix the way which I'm writing on the top and what I'm writing on the bottom, because they are otherwise mutually exchangeable. If this is an alignment, if I move this up and this down, that is also an alignment. That is clear. Okay, once you fix, let's say you fix this length I fix, okay, like this length is m, because m in this case is larger than n. How many possible ways are there to? Actually, I can. That's true, because I'm allowed to do that. That is correct. Yes, this is allowed actually. So it is indeed m plus n. That is correct. Does everyone see why it's m plus n? That is allowed actually. That's correct. Okay, good. Okay, now once you choose, right? So these are the total number of ways I can move things around. Let's say I choose. So, I mean, this is the total maximum length of things that I get. But if I choose a particular length, let's say I choose m, that I want to do an alignment of m, how many possible ways can I do this alignment? This kind of alignment, where my final length is m. How many possible alignments can I do of length m? This is one alignment. This is also again an alignment. Okay. Okay. So intuitively, what you're doing is once you fix the length of the alignment, your length goes from 0, right, to m plus n overall. 0 will basically mean that, sorry, this is a minimum of, not 0, minimum of m and n to maximum of m plus n. This is the length of your alignment. Once I choose a length, what do I choose? So my sigma, like there are a number of possibilities. So first, I'm choosing what is the length of that box that I'm going to draw. Once I choose the box, how do I choose how many number of alignments are there? Hmm? Oh, max of from? Ah, that's correct. Sorry. Yes, sorry. A lot of mistakes. Yes. It should be max, because you're bounded by the original length to be. That's correct. Yes. Max of m plus n to m plus n. So let's say you choose some box of size k, right? So your this box size, delete in what sense? Yes, you are allowed to delete, but this is the initial undeleted length, right? So minimum will be at least this length. You cannot delete, you can insert deletions, but I cannot delete T, right? I cannot change the sequence. You cannot change the sequence remains the same. You can insert deletions, you can insert dashes. That is correct. So max is correct. Let's say you choose a bound of box of bound K. Okay, so A, C, whatever, K. K is the entire length. And the other one, of course, by definition, since the box is of K, is also K. Okay, now it's a counting problem. This is the definition of some operator that we studied previously. How many number of ways can you do this alignment? You cannot also, of course, switch the order of the bases, right? You can only put them in the order. What you're allowed to do is you're allowed to pick where to put dashes. That's all you can do. So how many ways can you put these dashes? K is the entire rectangle length. It is minimum of max of m by n. How many ways? So you have to insert certain dashes. You can put k minus 1 gaps. But this length was beginning, this was big M to begin with, this was N to begin with. K is any chosen length of the alignment that I have chosen to take with a minimum of M. So let's take this example, maybe it will become clear. Okay, so A, C, T, C. Should have picked a smaller example. Hold on. Okay, let me pick a smaller example just so that I can flush it out. Okay, this is your M is 5 and your N is 4. Okay, how many possible alignments are possible is not just this, but I could also do starting from, I could do an alignment like this where there'll be all dashes here. So the maximum I can go is this boundary being M plus N. Okay, this is M plus N at max. I'm saying that you pick any K which is between M plus N and max of M by N. So you have drawn those boxes, and within those boxes, how do you arrange your dashes? How many ways can you arrange your dashes? The problem has started from there. So for example, let's just take when K is equal to 5. This is one alignment. How many such alignments are possible? How 5? Yeah, how 5? How 5? Correct, right? So 5 possible. Among these 5 positions, I can, in the N, I can choose one position. If I increase it by 1, very hard to arrive at a full answer, but we will see the argument. How many ways? K is equal to 6 now. We'll just see intuitively, will not solve it. Okay. How many ways? Among this length, I can choose one for my dash. And among this, how many? This upper bound, yes, there's a constraint, there's an upper bound. That's why I said solving the final problem is not as straightforward, but you get the intuition. Okay, it's not as straightforward. It's a counting problem overall. Okay, so if you actually want to just put an upper bound on it, that upper bound is for a given length of N alignment, you can choose your K to be the number of dashes that you have. That dashes will go from 0 to min of M plus M by N. It's exactly the reverse of going from here. If you subtract min from this, that is the bound that you get. You're just choosing the number of dashes that you have to put. It is equivalent to N plus M min choose Mn, right? Intuitively, is this idea clear? What we are doing? So this is exponential, right? The number of alignments, possible alignments that you can get is exponential. And that is what you should first appreciate before we do the rest of the stuff. So in this example, what we did is we are picking up how many number of matched pairs do I want to have. Here I showed you the example using uh, uh, dashes, and when I actually, the simpler way to solve this is when you pick how many number of bases can they align to, right? So the mismatches are of course allowed, but in this case, K is 1, 2, 3, and that's it. Yeah, mismatches are allowed for. Okay? Mismatches are allowed for, of course, in this example. So you can choose how many number, I don't know why this is happening. So you can choose how many number of alignments you have with perfectly K number of matched pairs. And the K number of matched pairs will go from minimum, right? So this will get flipped. Minimum because you can only at max have n pairs match, because you have n is your smaller number, for example. And max of it could be, excuse me, the minimum number could be 0 and the maximum will be minimum of m by n. You are essentially pulling out how many number of matched pairs can you align and totaling it up from 0 to min of m plus n. At each point, you're choosing among the m entries which k base pair should I choose, which k base pair should I choose, which should map, and that's why it becomes mck times nck, which is the expansion of this binomial coefficient. Yes. You're not allowed to keep these dashes one by one. There is a edge effect in this case, which it is not accounting for. That is correct. Yeah. So I'm not subtracting that. This is the upper bound in terms of maximum possible number of alignments. I'm not subtracting that explicitly in this version. Okay. This number, if you calculate for any m of m, you'll see that this is an exponential hard number. And that's why we want a strategy which will help us come up with a much faster way to calculate what is the most optimal alignment. Okay, everyone with me? Okay. So what does global alignment involve? So global alignment is given two DNA sequences. We want to align them end to end. So they should be aligned end to end, for example, in this case. And each base is sequentially or consecutively, we select each letter and try to find out what is the best possible placement for that particular letter, so that in the end, the entire sequence is well aligned. Okay, so one way to think about the sequence alignment problem is thinking of this as a graph. For example, you have two sequences of length 4 and 3. If I put them, again, the order does not matter whether I put one of them in the rows or the other one in the columns. Essentially, what sequence alignment is trying to learn, for each base position that I take, this is like a chess square board. Your goal is to reach till here if you're doing global alignment. And how you reach there by putting one base at a time tells you how did you arrive at that final location, which gives you this alignment to be the optimal alignment. The idea here is that each step that we take, we can calculate a score, and we'll see this in just a bit. The goal is that we try to optimize each step's score that we get for each of these cells. And in the end, we can calculate what's called an alignment score. If I take this path on this graph, the final score that I get along this path will be the maximum. That's how the algorithm is designed to be. Is this rationale clear? You're starting from these dashes. Dashes means that you have no prior commitment to any particular base being aligned. You start literally from two deletions. You add one base at a time to each location. It doesn't matter whether you go this way or you go this way. We'll see how the scoring is done in just a bit. But we keep iterating and we find out what is that path at which my score that I get of alignment to each single base is maximized. Okay, this part is clear. We'll see the scoring strategy and hopefully that will become clear, right? So Imagine you had two sequences of length m and n. There are two possible ways, right? One is you have these sequences which are up to I base and j base, right? So they're not equal in length. There are three possible cases. Your I and bj minus 1, this is what it looks like. At the ith position, at the jth position, I'm aligning bj to a deleted base. In case B, I'm aligning AI, the ith position, to a deleted base, or in the case C, I'm aligning AI to BJ. Mismatch or a match, deletion in B, deletion in A. Okay? Only three possibilities exist if I'm at the ith and jth location of a string. Is that clear? Sure. So you have traversed so you are given two sequences, A1, A2, AI. Okay? B1, B2, Bj. I'm at the ith, jth step. Okay? Think of this as rows and columns. Which one is where doesn't matter exactly. Okay? There are three possibilities now with the ith and the jth location. What is the first possibility? That this is case C, which is I choose to align the ith base to the jth base of sequence B. The second possibility is I choose not to align to Bj because that won't give me the optimal score. I choose to have a deletion here, which is your case B. And the third possibility is I take my Bj and align it to a deleted sequence, and that is your case A. Only three possibilities, right? At each single nucleotide that you're picking up, you have three choices. You can do a mismatch or a match. You can insert a deletion in second sequence, or you can insert a deletion in the first sequence. Size of? No, so this is not dependent because I am actually at the ith and the jth location. So I can be anywhere in my first string and I can be anywhere in my second string. How is the choice determined, right? So what do you think determines which path should I take? There are only three cases. Is that part clear? That part should be very important. It's very important to be clear. There are only three cases. Which case should I pick? What is my goal? My goal is that whatever step that I take, I get the maximum out of it. To get the maximum out of it, you have to tell me whether the mismatch score or the match score that you get is that higher. If I align it to a deletion, is that higher or is this higher? And that is what will determine me which path should I take of the three possible cases. Okay, optimization problem overall. Is that part clear? So we can define Sij, which is the score for taking for aligning the ith and the jth sequences, the bases. We can assume that S is the optimal score to I minus 1, j minus 1, which means that till here you have aligned and then I'm aligning AI and Bj, which is what this case is. Or you can assume that in the previous case, in this case, now I'm going to insert a deletion because this gives me higher score. Or you can choose this path, which because it tells you that this is the score that's going to maximize your overall score. In other words, you can write this particular part mathematically as if this is the score for aligning x to y at the ith and the jth location, this is the maximum of what the alignment looked like till the previous basis, and then you align xi and yj or ai and bj, they are same. Or in the second case, this is I minus 1 and j, which is your this case, where you introduce a deletion in the second sequence, or it is you insert a deletion in the first sequence. So the score that you get at a particular cell, and we'll do this as an example, and all of it will become very, very clear and very, very intuitive. This is how we arrive at the most optimally aligned sequence, because at each step, I'm trying to maximize among these three choices. That's all this step is trying to do. Okay, is that part clear? This is called the Needleman-Wunsch algorithm, developed in 1980s. So not extremely old, also not very hard to understand, right? Is the general idea clear? Okay, so this runtime of this particular algorithm is O of n square, which means that if you run this on 2 DNA sequences, it basically might not finish in this lifetime. Right? It's 10 to the power 18 overall. If one comparison, depending on how much does it take, if it take 1 second, you have 9 into 10 to the power 18 seconds for this particular computation to finish. But we know that we can easily compare 2 DNA sequences. So of course, this is not the algorithm that we require, but this was one of the first algorithms that came about to be for solving this sequence. Now there is one important step, and all of this will become clear in the next slide, is how do you initialize, right? So this paradigm of problems is called the dynamic programming paradigm. Why dynamic and why programming? Well, because it's used this kind of structure about using information from the previous step to optimize the next step. It makes it dynamic, so it depends on your previous step that you took. One of the things that you have to do for any dynamic programming algorithm is to have certain initialization conditions. So I'll give you the intuition of how to initialize this. Is any questions about anything on this slide? We will do an example and it will become clear. But let's say, any questions? Yeah. This is the length of the sequence. So imagine you have 2 sequences of length n. This will take O(n) square computation to finish. Yes, but I'm going all the way from 2 lengths, right? So I and j are at at the jth step, but this goes from ai to an and b1 to bn. So to come up with an optimal alignment for this, I will have to do O(n) square. I'll have to take O(n) square steps, right? And also memory actually. Okay? Question or observation? Clear? Okay. You have seen one version of this in no. In DSA, yes, right? Okay. Do they teach Needleman-Wunsch in that class? Okay, for sure. Lead code, does it cover Needleman-Wunsch? Similar-ish structure for comparison, right? So, for example, when you type in Word, right, you do Control F. This, of course, you cannot run because if you run this, it will never finish. There is a smarter version of this, which we'll see if we want to cover this in this class. I usually don't cover it because it totally takes it into a different tangent. There's something called the Z algorithm, which is used for much faster string matching. But you can think of so why do we care about this, right? So this is basically a string matching algorithm, right? So even if you're not interested in biology, you're basically using ways to figure out how to align 2 strings. Okay, how do we figure out where are we coming from? How do we figure out the alignment? We keep track of the path that we took and we backtrack how we arrived at that location. This is one of those classic cases where I should show you and not tell, okay? Very simple example. Sorry, I was talking about the alignment problem, the initialization problem. So when you initialize any dynamic programming algorithm, you have to set up the condition. So let's say you had these, I want to align B1, B2, any random sequence to A1, A2, A3. Give me the best possible alignment of the sequence, which maximizes the overall score. When I begin, I add a slash or a dash to each of these sequences, which says that when I begin, I will align a dash to a dash, and that should not incur anything. That should be a 0 score. If I go here, right, I'm moving B1 to B2 to B3 to B4. But here, here I'm going to say that what is the score? This is how I'm going to initialize. So here I'm going to say that if you align B1 to a dash, it should incur a negative penalty. because usually aligning deletions is not a good idea. This will be -2, -3, -4. What does this alignment look like? This alignment looks like you seriously want to discourage. This line is that alignment which you want to discourage. So in order to initiate that, you initiate your, and the same thing you can do here. And this number can be anything, right? This factor will be given to you, how much penalty does it incur. But you want to discourage alignments like this, so you initialize your conditions like this. That if you align anything where no alignments have been done to a base, to a dash, which means to a deleted sequence, it should be a negative, overall a negative number. I can swap these numbers to be anything. I gave you an example, but these numbers can be anything. This will be given to you in the problem. And you'll do an assignment problem where you'll get a sense of what happens if you change the deletion problem. But this number ideally should be negative because you want to maximize your score and you want to discourage things like this from happening. Okay? Clear? Sure. What's your name? Last bench. Nirav. Okay. Everything clear? Okay. Okay, let's do an example. So you're given a particular problem, very simple problem. You want to align what to y. Okay. If you match, for example, if you align W to W, you get a match score of 1. If you do a mismatch, so if I align W to H or W to Y, I get -1. And if I insert a deletion, insertion or deletion, that gets a score of -2. I have already initialized this condition for you. So -2, this into -2 into 2, -2 into 3, -2 into 4. Okay. Now what do we do, right? So what we are going to do now, I should draw this on the board. We'll do this example and then you'll solve the next example for me. Intuitively, you know what the answer is, right? You know what should it. So I want to align these two strings and I'm going to take a complicated route to it. So this is 0, -2, -4, -6, -8, -2, -4, -6. I will go to flash this in front of you and you are going to tell me, 1 second. So your SAI BJ is 1 if AI is equal to BJ. It is -1 if AI is not equal to BJ, and your AI dash is equal to okay. Now help me finish this matrix. It's a matrix completion problem now. So I'm here now, okay? I'm here. I already said that this is the score that you're going to get if you align what to all dashes. This is the kind of score that you're going to get if you align y to all dashes. Okay. What will be my score here? Okay, y1. Say that again, sorry. Okay. And okay, so but it is coming from somewhere, right? Where is it coming from? 1. Which of this scenario does it need to hold for that 1 to happen? Okay. So whenever, is this clear? 1, you're aligning W to W, AI, BJ, this happens, this score is 0. I cannot forget this score because that's what my dynamic programming is. So you will mark it like this. Okay, that you came here directly by matching the score. Essentially, that's what it implies. Let's do one more. Let me go here. You can choose how you want to go. You have to fill the entire matrix, but you can choose whether you want to fill it this way, or whatever way. I want to fill it this way. Okay, why -1? Yes, right? So I'm here. I can either reach from here to here. If I reach from here to here, I am aligning, I'm going to align H to W, which I can do, that's fine. I will get -1. So this would be -3 if I did that. So I'm going to write this possibility as -3. I could have also come down from here, which would mean that I'm aligning I'm making a deletion in the other part. So I'm aligning H to a dash. And that would have, what would be the score there? -6. And finally, I can go from here to here, which means that I will align my what? What is this route? Okay, so let's list down the routes, right? So in this case, my W was already aligned to dash. I aligned my H to W. So can you see this? Sorry, is this text visible? WH dash W is this dashed route. What about this route? You need to always make sure where you came from. So W I had already aligned, and now H to dash. Okay. Here, what should I write here? WH dash 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 W. Okay. Anyone this is not clear, please stop me now. So if I do this, right? So when I do -2, I'm saying that this is highly discouraged. When I said -4, this is also discouraged, because I would have come, only way I could have come is from this direction. This is also discouraged, and this is also discouraged. This is -2, -4, -6, -8. You're initializing because there is only one way to come here, right? Which is this line. Similarly, this line, there's only one way to come there, which is dash 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 y. Right? So -2, -4, -6. Okay, that is clear. Now, what's not clear? Okay, take it, fine. Okay, let me slow down a bit. Okay, I'm here. Okay, which means that I want to see how can I reach here? Like, how do I reach here? This is a graph. You're traveling the graph, right? You can either take this route, or you can take this route. You know intuitively, don't forget, you know intuitively that W should get aligned to W, but forget about that. In this case, what is happening is whenever you take a diagonal, that means either you are incurring a match, a full-on match, or you're doing a substitution. This is case A. Here there is nothing, so dash is aligned to dash, that's why it gets 0, that gets deleted. Okay, so if I align W to W, this condition is satisfied. So I have to, so I don't want to write this again, but this is xi minus 1, yi minus 1 plus s xi yj. Right. So this is 1 because that is 0. What about this? You're already at -2, okay, and you're moving in this direction. So which means that you are not making a mismatch, you're not making a substitution, you're taking the deletion route. Okay, so this is, sometimes I write it this as B and A. Again, the thing does not matter, but this is your B sequence B and this is your sequence A. You can also flip it, doesn't matter. If I'm doing this, right, what is it that I'm saying? So I came from here, I went down, so I said I was here, this to this is 0, So I start with this and W. And now what am I doing? I have W now. I have come to W. And I'm saying that you came from here, which means that you aligned to a dash. You would have to align to a dash to come here. Okay, which would be what? So this is -2 there, right? So 2 -2. You already had a minus score because you had aligned this. Now you're forcing W to also be aligned to a dash. So this would be, so I will write the first score as 1. Actually, let me use different colors. -2. So this is -4. Okay. And what about this one? What about this one? So now I have W, right? So this, so yeah, just let me just delete this. But you aligned W to what now? Correct. And what is the penalty that you have to incur because of this? You were already at -2, and now you will have to have another -2 here in this process. So there will be -4. And max of this is 1. So that's why it shows that I'm not going to take this route. I'm not going to take this route. I'm going to choose the path of maximum score, which is 1. That's how I got 1. Okay, clear? This is what is happening downstream for people who have run Blast. Some version of this runs Unside Blast. We'll see exactly what happens, but this is the actual algorithm that's running besides your any of your toolkit. Okay, now this, if this logic is clear, you should be able to help me finish the rest of it. Okay, here minus 1 second, -1 from where? From the left. So 1 -2. If I had taken this route, I would have been -2 and -1, -3. If I had taken this route, -4 -2 will be -6. 1 is maximum. I'm going to keep this arrow because I want to know where I came from. This one, -3. Again, I came from here. That's the best possible score that I can get. And then -5. Okay. Any doubts, any questions, anything not clear? Simple logic, right? No hard math, no statistics, very simple logic. At each step, I only have 3 possibilities. You can either come from this direction, you can come from diagonal direction, you can come from this direction. You're coming from this direction means you're aligning W to a dash. You're coming from this direction means you're aligning this from W, and so on for that particular base. And if you're coming from this direction, means you're forcing things to align, which are XX are 2 different bases. Okay. So this direction substitution, this direction deletion, this direction deletion in A, this direction deletion in B, or vice versa, depending on how you write A and B. Okay. This part is clear? Okay, quickly now, let's finish the rest. How do I reach here? Somebody who is not Ashwajit. Okay. Here, how do I reach? I can reach from here. I can reach from here. I can reach from here. Which one should I choose? A 0. Okay, so forget about this one. It's always I minus 1. So you already reached here, right? That's how you got the value. So we'll just focus on this. Okay. So 1. Okay. Now here. This one is simpler. How do I reach? How do I reach here best possible way? I go from here to here will be a deletion. So -1 -2 will be -3. -1 -2 -3. 1 plus 1 will be 2. Okay. And I will have to keep my direction that I came from here. Okay, this one. I can go from here to here. So 2 minus 2, 0. Go from here to here, -3 -2 -5. -1, force it to mismatch, -2. Okay, here. What did I do? Ah, okay, correct, right. Sorry. This is 0, correct. Good. Okay. This one? -3, -1, -4, -5, -2, -7, 0, -2, -2. Okay? Okay, last one, almost there. This one? Minus? Okay, we came from down from here. Louder? 0. Okay, correct. This one? Okay, now where did I come from? Where did I come from here? Where? Here? No, I don't think I came from there. I can either come from here or I can come from there. Doesn't matter. We'll see what it means. Does everyone see why? Deletion here or deletion here. We'll see what it looks like in just a bit. Oh, is it 1? Ah, correct. Okay. Sorry, then this doesn't hold. Okay, assignment problem, different scoring function, so you reach there irrespective. What did I do actually? I have. Okay. Okay, final. Okay. Now? Okay, now where did I come from? Can I come from here? Clear? Anyone, even if there is a single person, this is not clear, will not be able to do the next assignment. So please ask me now. Not today's assignment, but the assignment that comes out next week. Anything that's not clear? Needleman, Wunsch. You can code it, very simple to code. I will give you the boilerplate and you will see how beautiful it looks, what is the time complexity and so on. Okay, so what does, like, how do I figure out what is a alignment now? I'm going to do what's called backtracking, right? So backtracking essentially involves me going back. So this is my final score that I get. I will backtrack and you will create a data structure which allows you to do backtracking. You will start here, okay? Now there were two possibilities that were happening there, right? Either I could have aligned my A to Y. This is, I'm going to call it 1, or I could have done what? Louder. Possibility 2 is what? Something is wrong. Let's see. Is that correct? Correct. So whenever you're taking this diagonal, which means you're forcing the mismatch to happen, right? So this is correct. Second one was not correct. So what is there in second one? You're going from the same base, right? You were at base A and Y, and you're forcing a dash in what? T, right? So this direction means a deletion in this sequence. Okay, don't remember it with A and B, and that's why I don't highlight, because this can of course be flipped if you're going horizontal, right? The way to understand this is if you're going horizontal, you are already at the last base. So when I'm picking up this base, I'm forcing that to get aligned to this dash. So horizontal deletion in the second sequence, or the sequence along your row, which is also like this, right? So maybe that's one way to remember it. Don't have to remember anything, you just plot it and you'll figure out. Okay. Okay, Fine. So what should I do next? I am here and I know in path 1 I came from here. What did I do here? I came to A from this direction. Okay? Is that clear? If this is clear, this should be clear. Okay? Now I'm at A. So I'm here now, sorry. H with H because you know you came from here. And you know again you came from here. So W to W. Okay, let me finish. Second one. So second one, we came from here. We are now here. Okay, right? AY. Again, you took this route. Whenever you take this diagonal route, that means you're doing a mismatch or a match. In this case, it happens to be a mismatch. So now you're here. Huh? Question? Now you're here. Now where? Same. That follows, of course. And that gives you two possible alignments with this scoring strategy. Very intuitive, right? You already knew the answer. This is just formalizing it. That's why the slides say formalizing it. Okay. So we went much slower than what I had thought, but we will. Let's do this as an exercise. Okay. So you have ATC GT and AT and TGG TG. So I'm not going to show you the. I'm not also going to do it. It's slightly lengthier. ATC TG and TGG TG. Mismatch is match is 1, mismatch is -1, and this is called gap penalty or an indel penalty, that's -2. You'll do the same thing, which is going to draw this bigger square. You only have to do it a few times to get a feeling of it, and that's the only reason that you should do it. In a modern world, you never have to do this, of course, right? You will use computers to do it, but how to get a feeling, and the reason to get this feeling is because what we are going to do next requires that intuition to be clear. Okay. Any thoughts about intuitively what it would it look like? Yeah. Okay. This you can slightly cheat, huh? Like so A T C G T and T G G T G. T G G T G. It's a pattern matching. Okay. What should I do intuitively? Shift. Yeah. So T here, G GT G. Okay. Maybe sometimes your intuition is right and sometimes it is not. I think in this case it will be right because the scores are in that favor, right? Remember, this score has a meaning. It has a biological meaning. Matches are always good. Mismatches are okay, but deletions are bad, right? Because we know in biology, if something is getting removed, it can have much stronger consequences as compared to mismatch in most cases. So do this as an exercise. Of course, the solution is available. Don't look at the solution. I have 2 minutes. I will motivate a problem. What we discussed is we talked about global alignment where we were forcing the end of the sequence to align to the end of the sequence. However, there are cases where that is not required, right? Particularly, for example, if we were aligning that entire length, the entire segment of the virus DNA sequence to the entire human gene, that is not required because the entire thing will never align. What we want is there a segment in the virus genome which aligns with some part of my gene segment? And this becomes what's called a problem of local alignment. This is something which is close to my heart because when I decided to go for PhD, this is the only thing I knew in computational biology. This algorithm that we're going to talk about was developed by Michael Waterman. Michael Waterman was the reason I chose to go to the school that I went to because he was a professor there. He is a mathematician. He's a full-on probabilist who happened to chance upon biology. And then what he, along with working with a biologist, this is famous algorithm called Smith-Waterman algorithm that was developed. Smith was the biologist. Waterman, of course, is the probabilist. He started the computational molecular biology program at USC, University of Southern California. And that was my only reason to go there because that's the only thing I knew about computational biology broadly. But he had retired by then, and unfortunately, he was around, but he was not really taking students. What was fantastic to be still around people like Michael is the breadth of knowledge that he has, which I don't think ever in my life will I be able to capture. He's also a wonderful writer. So if you look him up, he has written multiple books. A lot of these are memoirs about where he grew up. He grew up in a farm, by the way, in the US. Worked at Los Alamos, which some of you might know for other reasons. And then just was doing probability theory when this particular problem from molecular biology came. This is exactly the problem that was given to him. And the way he described me, and hopefully I'm able to do justice to it, is when they started looking for this particular problem, solution for this problem, that human genomes are coming out or partly coming out, and they want to see that where exactly are things which are matching, they would print these DNA sequences. This was the era of dot matrix printers, which I think maybe in this class nobody would have even heard about. Anyone has heard about dot matrix printers? Okay, wonderful. So dot matrix printers are dot matrix printers. They print, each word is printed as dots. They would print this on long sheets of paper, sit down and try to move the paper so that the sequence aligns. And that's where the discovery of this algorithm happened. And when I show you what the change is required in global alignment, you'll be surprised. It's such a simple change. But that took few years to crack. And finally they had it. It became a big, it's one of the very well cited papers in molecular biology. Of course, there are other methods as well, which we'll talk about. But that's how the birth of local alignment happened. So is the problem clear what we are trying to do, local alignment? I already showed you the scoring function. Any ideas about what will you change in the scoring function? If not, we will start this class because we are actually out of time next this Thursday, talking about what the simple change is. I will also, on a related note, I will also encourage you to think about another problem which is called the Columbus egg problem. Anyone has heard about the Columbus? Anyone has heard about Columbus? Columbus people have heard about, right? Who's Columbus? Christopher Columbus. Discovered, finish, America, right? The great land. But when he went back to Europe, he was like, there were like people like, this is not a big deal. It's fine. Like somebody would have discovered it at some point. What's the big deal? So he said that, okay, I'm going to give you an egg, and can you put it flat? Can you make the egg stand flat? That's the problem. If you can do it, I will believe that you are as good as what I am, or whatever I did is not trivial to do, even though somebody would have discovered it at some point and people had maybe visited America at some point. Nobody had actually discovered it. There is something which is non-trivial about what I did, and he used the example of an egg to show it. Any idea how would you put an egg flat? If you put it, it is always going to, it's elliptical, so it's always going to roll. Any ideas what you would do? Wow, we have a Columbus here. Okay, that is exactly what, that is the right solution. So you'll bump it at the bottom. Nobody could do it actually. None of the Europeans could do it. Simple question, simple approach, right? Just bump it at the bottom, and that's what the Smith-Waterman algorithm is about. Just making a small bump in the algorithm that already existed, but was not the squared. Okay, I'll stop here and we'll resume from this point in the next class.